All right, good morning. Welcome to my least favorite part of this workshop when I'm talking. And it will be followed with my favorite part of this workshop, drinking coffee. So before we start, just like the students, we had a meeting yesterday, the teachers' meeting, and then they asked for actionable hobbies. So do you think photography is actionable? Let's see how many people think it is not actionable. There you go. And there was another guy yesterday who said it's not actionable, and now I just put an action on it. Right? So now he's involved in my photography. All right. I'm an assistant professor at Bilkent University in Ankara, Turkey, and I work in genome structural variation discovery using various sequencing technologies. And today I'm going to talk about some of those methods available in the literature, and this is supposed to be a tutorial talk. Therefore, I'll just try to cover the algorithms themselves, and not necessarily mine. And I'm also supposed to, suppose that none of you have any background in this. So we know that the genetic variation comes in different sizes. So the smallest ones are single nucleotide polymorphisms. It is one character changes. There can be small insertions and deletions and microsatellite expansions and contractions up to 50 base pairs as defined by the 1000 Genomes project. And the larger ones are called the structural variants. And again, this can be deletions, insertions and insertions of novel sequence, or insertions of mobile elements. Also segmental duplications, which are duplications of size larger than 1 kb, copied to another part of the genome with a sequence similarity more than 90%. There can be inversions of some DNA and translocations of some DNA from one chromosome into another. And there are much larger ones, the chromosomal changes, where the most well-known is the Down syndrome. So it is the third copy of chromosome 21. So collectively, these ones, the deletions, insertions, and duplications, are also called CNVs, which stands for copy number variation, because they change the amount of the DNA content. So basically, these are the structural variation classes. These are the most commonly known structural variation classes, where again, some portion of the DNA is deleted from the sample, so that pink part is deleted from the sample. I don't know if this works. This doesn't work. And there can be some new sequence inserted, which has no other representative in the reference genome. There can be mobile element insertions. So these are short sequences of usually bacterial or viral origin that were integrated into primate genomes millions of years ago, and they still copy-paste themselves. So we do have 1 million copies of ALU, for example, in the reference genome, and their location might change from person to person. Most of them are fixed, but there are still a handful of them that copy-paste themselves. Segmental duplications can be either in tandem orientation or interspersed. So they are right next to each other, or they can be copied to some other part of the genome, and this can even be in a different chromosome in the genome. There could be inversions of DNA, where the DNA is flipped at some location, and there is translocation, copying from one chromosome to another, which is common in cancer. So this is again just copy number variants, and these are called balanced rearrangements, because the inversions and translocations typically do not change the amount of the DNA in the cells. So why do we care about structural variation? One thing, it is common. So this is from a very old study, 10-year-old study published in Nature. So this study analyzed the genomes of 9 individuals and found insertions, deletions, and inversions. So these are relatively large scale, more than 5 kb each. And you can see that they are scattered all across the genome. So they are individually rare, collectively common. That's what we say them, because we expect about a couple of thousand of structural variants compared to about 3.5 million SNPs. But they also affect more of the DNA. So the total size of the structural variants per person is usually on the order of 20 million base pairs. Some parts of the genome is harboring more of those variants. This is typically because there are more segmented duplications and more repeat content in those regions of the genome. So it is common, and they also might cause disease. So there have been many studies over the last 15 years or maybe even 20 years that associate copy number variants with disease. So this is basically what happens. So let's say on the top left we have two chromosomes. So during the meiosis there is a crossover event, and let's say we have those green boxes as segmented duplications. So they are highly similar to each other. Since they are highly similar to each other, during the meiosis event they might cause an unequal crossover. So they just misalign. The wrong copies align with each other, and then when you switch the DNA, and one of the gamete has the third copy of the green box, and one of the gametes is missing one green box, and also one of the gametes have now this extra sequence of ABC. So now you have a secondary duplication in one of the gametes, and in the other one you have a deletion. And if this happens in a functionally important gene, this might cause some disease, and so again some mental retardation and autism and schizophrenia cases were associated with CNV. And yesterday you listened to Feridun when he was talking about these variants happening in non-coding genomes of the non-coding portions of the genome, yet still causing disease. So in the case of segmental duplication, so even when you are not losing a gene, having an extra gene might also cause a disease. So there are here are some examples here. The first one is a deletion, but the second one of the defensin gene family. They play a role in the immune system, and an extra copy of the defensin gene family is associated with psoriasis, for example. On the other hand, if you are losing a copy, this is associated with Crohn's disease, so it goes both ways. An increase in CCR3L1 is for HIV resistance, increases resistance, and increases the susceptibility to HIV. So let's talk about 10 years ago again. So this is my favorite quote in the field, I guess. So this is a quote of a day in 2008. So this is authored by Elaine Mardis, who was the co-director of WashU Genome Center back at that time. And this was during the first 1,000 Genomes project meeting. And this is the exact date. I actually checked that yesterday. So she said this. So yeah, SVs are fun. They are very difficult to detect, but this part of the fun. So let's talk about how to detect these. I mean, I used to have a slide of discussing the older technologies, but since they are all dead, those arrays and such, I'm going to talk about whole genome sequencing. So we start with a test genome. So let's say we are analyzing Feridun's genome, and we get his blood, and then we randomly shear it into pieces. And obviously, when we get his blood, we are going to have multiple copies of his DNA. And then we apply some size selection depending on the technology, not necessarily for all technologies. And then we can do pair-dance sequencing using Illumina, which we are going to have short reads. Or we can do single-end sequencing using the newer PacBio and nanopore technologies. And then we map them to the reference genome with your favorite aligner. Typically, this would be BAA for Illumina and Blazor for PacBio, or NGM LR, the new one. So this is basically how it looks. We start with the sample, and then we randomly shear it into pieces and randomly just read small portions of the DNA. So this example is for single ends, but you're going to get the idea. And here we actually have two sources of changes, two sources of errors in alignment. One, of course, we don't have the sample's DNA, so Feridun's DNA at the top. And when we are comparing with the reference genome, so there will be some differences. So those blue font, the blue colored characters are the differences between the sample and the reference. But of course, we don't have the full sequence of the sample. And at the same time, the sequencing technology also makes mistakes. So those red font characters are the mistakes by the sequencing technology. So for Illumina, this is about 0.1%. and for PacBio, 12%, nanopore 20% per read. When we have long reads, obviously, we are going to see actually more mistakes made by the sequencer. So now we have those we sometimes have extra characters. So for example, the last one, the second last one from the bottom, it has some extra A's which is not actually in the sample. And there are some portions, so the third from the bottom, it has some sequence missing. So we have more insertion and deletion errors. So here with Illumina we have more substitution errors, but with PacBio and nanopore we have more insertion and deletion errors. And a more recent technology, link reads, is actually using Illumina technology for sequencing. So it has the same error profile, 0.1% substitution errors, but you can also link some of those reads together, which says basically that they are coming within 30 to 50 kb of each other, typically, and they are also coming from the same haplotype. Obviously, for human genomes, we are diploid, so therefore some of those are coming from one haplotype, some of them are coming from the other haplotype, which also contributes to this error, right? When you have heterozygous mutations. Here we can say that they are coming from the same chromosome and within some distance from each other. So with the link reads, we typically have 2 or 5 colors, well, 2 to 5 molecules that are colored the same. So here we have the top one and the fifth one from the top colored the same, so they are all red. So we are going to say that they are related. But so since in this case our sample genome is so small, they are overlapping, but in reality they will not be overlapping. So the maximum number of molecules that you are going to get with the same color is going to be 30. The typical will be about 5. So back to those sequencing technologies, we have the short read sequencing technology, Illumina. They produce short reads, paired length. We have billions of them. They are relatively very accurate. We have the long read ones that can be up to a megabase with nanopore, but they are single end when they are indel dominated with a lot of errors. And the long range sequencing, so which is the link read technology, it is offered by 10x. So it is an addition to the Illumina platform, therefore the sequence properties are the same, but we have an extra information of this range. So using those, there are a lot of algorithms developed, and an algorithm is published every 5 minutes. So there should be about 3 more papers since I started this talk. So I'm sure I am missing a lot. Maybe you also developed something I'm missing. I apologize. So these are some of the examples for Illumina. We have TARDIS, DELI, LAMPI, breakdancer, Pindel. Nanopore, we have Lava, Smart SV, Cineples, and HISA. And Lava and HISA actually use both Illumina and PacBio together. And for long range, we have Valor, Grok SV, Enable, Long Ranger. So the ones written in red are developed by my group in collaboration with others, and obviously they are the best because they are written in red. So back to the whole genome sequencing. So we get those sequences and then we sequence and we map them, and we are going to look at the properties of these mappings to figure out the structural variation, right? So basically we can use 4 different sequence signatures to detect these structural variants. We can do read pair analysis if we have read pair information. For example, on the bottom, we have the read pair coming from Feridun, and let's say he has some deletions somewhere. And we are going to know the approximate size of that molecule, because we are going to have about 1 billion of them. And then when we align, most of the genome is not going to be structural variant. Therefore, you can calculate the average size of those fragments, and then the standard deviation. And if it is too big or too small, then it's going to be an anomaly. So if it is too big, so let's say the average is about 500, And then when you map them to the genome, they are mapped about 5,000 base pairs from each other. So this is going to tell you that there is probably a deletion of size 4,500 base pairs. Obviously, you are going to require more evidence than just one single read pair. So in theory, you can find all forms of structural variation, theoretically. In practice, doesn't really work that way. And the size of the variance that you can find and how close you can get to the breakpoint, so these are the actual locations where the deletion or the structural variation starts and ends, is going to be dependent on the size of those fragments and how tight the distribution of the size of those fragments is. You can do read depth. So this was actually the first method developed back in 2002, way before high throughput sequencing. So again, you just take those reads, map them to the genome, and then you just count them in a sliding window. And if you see a depletion of reads, that's going to signal a possible deletion. If you have an increase, it's going to signal a possible duplication. You can do split read analysis, so which for long reads, this is what we do, what everybody has to do. Is basically you are trying to align, and then you are looking for breaks in the alignment within the read, right? So in the read pair, you are looking at the distance of fully aligning two reads, and with split read, you are looking for a particular alignment, and then looking at breaks in it. Again, theoretically, you can find all kinds of structural variance with exact breakpoint resolution, again, theoretically. And the fourth one that you can do is local or de novo assembly. You can also do this also works better with long reads. What happens is you take the reads, you assemble them into larger contigs, and now you are going to ideally have much longer contigs, millions of base pairs, hopefully. And then you align them to the reference genome, and then you do something similar to the split read, right? Apart from with the split read, where you can have about 10 KB reads, ideally with the de novo assembly, you are going to have megabase size, contigs, much even larger. So the 1,000 Genomes project was a great opportunity for us to develop a lot of those tools, and actually a lot of those tools that we used in 1,000 Genomes project phase 1 are dead already, right? Because the technology has changed. There is long read sequencing, even the Illumina sequencing improved. So they are either dead or largely replaced with their next versions. So in the phase 1, we used 19 different algorithms for structural variant, and these are the list of those tools. So when you look at their detection power, and when you take only 14 of them, not 19, 5 of them were assembly based, so this excludes them. 6 algorithms were based on read pair, 4 in read depth, and 4 in split read. And then you look at the deletion calls only, and those deletions that are actually validated, so these are real, no false positives here. And you compare them, and you don't see much of an overlap between different sequencing signatures. So with read pair, you have about 7,000 extra, but with read depth, you have about 3,000 extra, and so on. So this is, again, because of the sequence complexity with read pair, especially with the 1,000 genome phase 1, when the reads were only 36 base pairs, mapping them correctly, accurately in a repetitive genome was difficult. Therefore, it works with relatively smaller size, structural variance with not much of a repeat content, and read depth works better for larger size CNVs, and obviously no inversions or translocations for read depth. And split read works for even smaller events that are in more unique regions of the genome. So what are the challenges? So again, we have genome sequencing, we have genome structure issues. There is high sequence complexity at breakpoints. That means we have a lot of common repeats. So these are the L2s, L1s, SVAs, ERVs, and so on. And we also have the segmental duplications, which increases the mapping ambiguity. And again, we have errors in those sequencing platforms. We do have GC, we used to have GC bias in Illumina. We still do. It's not as bad. So basically, when the amount of G and C increases, you are getting less reads from the Illumina platform. And again, for Illumina, the short fragment size and short reads provide little information. So this could be half width, long reads. So this is what happens. So let's say with the sample, you have those yellow boxes, duplications or repeats. And in the reference, you have those repeats as well. And then let's say in the sample, you have a deletion between repeat 1 and repeat 2. And when you have short reads that span that deletion, and you have those repeats, right? So the first short read may map here, it may map here, it may map to the repeat 3. And the second one may map here, and may map here. So each one of those possible mappings is going to give you a different sequence signature for a different structural variant. But when you have long reads, and this long read spans those repeats, so they are longer than the repeat length, what happens is that you are going to be able to do a more accurate split read mapping, and the first one is going to map to repeat 1 because there is some prefix that hits some unique region of the genome, so you can just continue aligning from there. And the second part is going to start from repeat 2 and end in the unique region of the genome. So you can use those unique regions as your anchors for better alignment. So let's talk about those sequence signatures one by one. So the read depth, as developed as WSSD back in 2002, used Sanger sequencing platform. So the idea was simple. So if you have a sequence to test, and then the reads are supposed to be generated randomly, so it should follow a random distribution. Not really the case, but let's just assume that it is. And then when you align them to the reference, so it will take a while. So let's say the black region is unique copy, the blue region is a deletion, and the red region is a duplication. So with the blue region, you won't see any reads mapping, maybe just a little of reads mapping. But with the red ones, you're going to see an increased number of reads. So this was the original idea. And then you count them in overlapping windows, and you just calculate your average read depth, your standard deviation, and then decide on your cutoffs, usually 3 standard deviations below and above. And if they are below, then you call for a deletion. If they are above, then you call for a duplication. So this was used pretty much the same way with high throughput sequencing as well. So one thing about the duplication though, especially for Illumina data, you should be able to track multiple mappings if you are interested in duplications. If you're interested in deletions, this is not the case. So this is what happens. So let's say we have a duplication of those red boxes over there, and then well, we have duplication of those colored ones in the genome, and then the reads are randomly generated from there. If you are using unique mapping, everything, let's say, maps correctly, and you don't see any change in the read depth, right? And if this is the duplication is the same with the reference. If you have an extra copy from the reference, you are still going to see some increase. And if you are looking at the multi-beam mapping ones, so those colored reads map to multiple places, then you see an increase in the read depth. But the read depth is assumed to be random. It's assumed to be Poisson, but it's not really the case. So these are really terrible. So these are from the first days of the 1,000 Genomes project. Fortunately, it's not as bad now. And you can see different distributions, or not really distributions, from those references. For these, we typically use some smoothing, statistical smoothing methods such as the lowest transformation to correct our biases. So back in 2009, we also published, so we used the same WSSD technology, we just adapted to Illumina with a new read mapper and further corrections in read depth. And then we analyzed the genomes of multiple individuals. Obviously, in 1,000 genomes, we analyzed thousands of them. And we looked for copy number polymorphisms, especially in the form of duplications. So the top line is the reference. Okay, it works. So this one is the reference genome, and so these are the duplications in the reference genome. Right? And then we have the genome of Craig Venter, James Watson, and 3 individuals from the 1000 Genomes project. And then when you align them and then you just count those reads, and then decide on your cutoffs, the green portions represent basically within 3 standard deviations of the average read depth. The red ones represent the high read depth count, which we call for duplications. Right? And then we have some polymorphic regions. So for example, this part is not duplicated in Craig Venter, but duplicated in everyone else. This is probably just a cutoff issue. You can clearly see the increase, but it just didn't make the exact number. And here we don't have a duplication in one of the trio. So this is actually a family. So this is a mother, father, daughter. So we don't have the duplication in the daughter and the father, but we have a duplication in the mother. Okay? So what we can do is we can also use what we call the single unique identifiers or single unique nucleotides, SUNs in short, to differentiate between different copies of duplicated gene families. So for example, let's say you have 6 copies of a gene in the reference genome. So let's say it is very common that most people have 6 copies. And then when you align them together, they are highly similar to each other, but there are obviously still some differences. And then you look at those base pairs that is unique in one of those copies. So those circled red ones are unique in representative copy, and there can be even some deletions or insertions extra in one copy. And the blue ones are not unique because they occur in multiple copies. So this G is in both copy 2 and copy 6. And then when you calculate the read depth the same way, just over those specific locations, and you require exact match, right, to those locations, then you can differentiate the copy number of duplicated genes. So this is from Emily's, again from 1000 Genomes. So Emily's gene family plays a role in starch metabolism. Amylase 1A family, they are expressed in saliva, and amylase 2 family is expressed in pancreas. So when you look at 179 individuals in this case, you see that in amylase 2 is not really polymorphic, but amylase 1 is really polymorphic with increased copy number in East Asians. So now we can differentiate those. So another example is that the CFHR and the opsin gene families, where we can say that in the case of the CFHR, an African genome NA18507 is missing 2 copies of the CFHR family. So if you just look at the regular read depth, you're just going to say there are 3 copies of this gene. But which 3 copies exist, which are deleted, now you can tell using those SUN markers. So for opsin, for example, again, the African individual is missing one of the opsin genes, but the YH, which is Asian genome, it has all copies intact. So the second method you can use is the read depth analysis. So again, we are looking at how close to the reads are mapping to each other. If they are too far apart, it is probably a deletion. If they are too close, probably an insertion. If they are mapping to some other location where you know that there is a transposon, so mobile element such as L2 or L1, it might signal a mobile element insertion. In this case, you are actually looking at both. So if that red arrow over there is the insertion location of that transposon, and now we are looking at both sides of it. So if you have a read pair coming from here and here, one of that, so they are going to map close to each other, but their mates are going to map somewhere else. Similarly for inversions, so these are the breakpoints of the inversion. So if you are spanning from the left side and the right side, they are going to map further away from each other and to different strands than expected. You can come up with similar formations for interspersed and tandem duplication. So again, there are a lot of algorithms. Variation Hunter was the one that we do with Feridun years ago. So we basically used a maximum parsimony approach. So our idea was looking at all the read mappings, what is the minimum number of variation that explain the mapping information that we get. So we were able to use all map locations. So this uses multi-mapping alignment tool. And then we basically just clustered those into potential structural variants, and then we applied an approximate solution to set cover. There are some other similar approaches such as Clever, Hydra, Breakdancer, and there is also again similar but with a geometric formulation called GASP. So this is what I told you about the size distributions. Again, most of the reads, above 95% of the reads are going to map within a distribution. So this is from a good sequencing library. If you look at the mapping distance between read pairs, you're going to see a tight distribution if you are lucky enough. And then you decide on your cutoffs. If it is too big, you go for deletions. If it's too small, you go for insertions. And of course, you also have to look at which strand your reads map to differentiate from inversions. It's not always as good. So sometimes you have distributions like this, which might be a problem. Definitely a problem for insertions because you cannot really cut off from the left side. So this is actually the read length. So the read length was 100. So anything below that is just overlapping reads, right? So it doesn't really tell you anything for read pairs. But for the deletion case, it is a little better, but you have a rather long tail over here. So our formulation back in the day, basically we defined the structural variation as 4 tuples. So for a given paired and read, we have the left mapping location, right mapping location, and the orientation. So this is the strand that it maps to. Plus minus for Illumina paired and sequencing, it refers to normal mapping. Plus plus or minus minus refers to inversion, and minus plus refers to tandem duplication. And then these deltas are cutoff values that are calculated from this distribution. So this is delta min, and this is delta max. We first cluster each structural variant signaling read pairs into valid clusters. So basically, structural variation is defined by a left breakpoint, a right breakpoint, and the minimum possible size of the variant and the maximum possible size of the variant. And we also have the paired end alignments. So in the interest of time, I'll just go quickly. We basically look at those read pairs that agree on the same type and the same size of the structural variation, and if they are in agreement, we call them valid. If they are not in agreement, then it becomes invalid, so such as in this case. Because in this case, we cannot actually decide on where the structural variation happens. So given those clusters, since we are also using multi-mapping information, we define each potential structural variant as a set, and within our universe, our elements are those read pairs, and read pairs may occur in different sets. Therefore, to minimize the number of variants, we use an approximation to set cover. So the third methodology is again split read. We are looking at the alignments themselves. So in this case, we have a break in the alignment, signaling a deletion. We have some extra sequence in the alignment, sequencing and insertion. So this alignment is broken into somewhere else, and then it is flipped, signals an inversion, and so on. There are a lot of split read based algorithms out there. The first one was from Kaye, published in Bioinformatics in 2009, Pindel, and then there are others. Strick was for the 454 platform. Maybe most of you didn't even hear about it. Doesn't really exist anymore. The 454 platform. We also have developed split read for exome sequencing in 2012. And for PacBio, you can use SmartSV, which also requires Blazor and assembler Kano. So it's not really just split read algorithm, but it also does assembly. And I'm going to leave the details to Mark on Thursday. And there is Sniffles, which is more recent. That is also developed for PacBio, which requires its own aligner called NGM-LR. And we have Mike Schatz at the back of the room, and he might talk about this in the afternoon. So you can also do assembly. So for short reads, we developed the novel seq, and then we extended into Pamir, and another group developed Poppins, which they all follow a similar strategy. Let's say we have a novel sequence insertion longer than the read pairs, and what happens is that you are going to have a bunch of read pairs spanning the left side of the insertion and the right side of the insertion, and only one end is going to be mapped to the reference genome, which we call the one-end anchored reads. And then those read pairs coming directly from the insertion. Since this is novel, there is no representative in the reference genome, they will be unmapped. So you take those, we call them orphans, you pass them through a de novo assembler. In this case, we use Abyss, which you can replace with any other assembler. And then we also clusters those one-end anchors in a similar way, in a way similar to variation hunter. So we are looking at agreements. And then we do a reference guided strand aware assembly of these one-end anchored reads, and then we have those orphan reads. Obviously we don't know where those orphans are coming from, but we know where these ones are coming from, and then we match those with these ones with suffix prefix matching. Again, build a graph and actually model it as a bipartite graph, and then just match those OAA contigs with orphan contigs. So again, in the interest of time, I am passing these fairly quickly. More recently, there are multi-signature SV callers. So now we are not using just one method anymore. We are using everything that we can. Smart SV is doing that, as I mentioned, it uses split reads and assembly. But for Illumina, we integrate read pairs, read depth, and split reads. So one of the first tools that did this was Lumpy. So basically, it uses those three different signatures separately. It first clusters the read pairs, then read depths, looks at the read depths, and then does the split read mapping, and then look at the potential structural variants, and then cluster them together using a probabilistic framework. More recently, together with Feridun, we also developed the TARDIS, which also is kind of an extension to variation hunter, because we can still use, optionally, we can use multi-mapping reads. If you don't have the time to remap your reads, that's fine. We can just use your BAM files. So in this case, we are clustering the read pairs and split reads together, and then use read depth as a filter. So the structural variants are hard, and the more complex SVs are harder. So this is the reported inversions by the 1000 Genomes project, the latest one, the greatest call set. And if you look at those inversions, and if you look at them closer, actually only 20% of those inversions by 1000 Genomes project are inversions. So the remaining 80% is something else. So they are more complex. So 54% are actually inverted duplications. I'm going to show you, why this happens. 7% had multiple deletions and inversions at the same place. 5% had a lot of variants occurring in the same place, and 14% were inversions and deletions happening together. So 80% of inversions are not really inversions. Well, they are inversions with some extra. So the reason is this. So let's say you have an inversion. So this is what you are looking for. In the case of read pair, when you have an inversion in Feridun's genome with respect to the reference genome, you have read pairs coming from outside the inversion, inside the inversion, and this is going to map normally, and then this one is going to map to the other side, right? And it will be flipped. So this will be our typical read pair inversion signature. If you have an inverted duplication, so let's say in this case you have a smaller sequence, duplicated and inverted and duplicated in Feridun's genome, and again you have those read pairs coming from here and here, and in reference genome you have a single copy. Therefore, this read is going to map way over there, and this read is just going to map where it is supposed to be, and then you end up with the same signature, right? So you have minus, 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 and they are separated. So this will be the same if there is a gene conversion event. So even if there is no extra duplication, if you already have a duplication in the reference genome and also in Feridun, And let's say there's some gene conversion event happening. So these differences, so these are the differences of those 2 copies in the reference genome. So those differences disappeared because of gene conversion. Again, it will be the same story. You're going to map this normally. The other one, normally, if there was no gene conversion, because of those differences, you would hopefully map them to the correct place. But since there are those differences are gone, you map that way over here. Again, the same signature, right? So if you have those cases, you have to call them as inversions. That's why 54% of those inversions are actually inverted duplications. Actually, the same story for deletions. Maybe we have a lot of false positive deletions in 1,000 genomes. So this is the same case. In this case, it's the duplication is not inverted. It's in direct orientation, but they are showing the same signature. So to help solve this, we developed TARDIS. I'm red flagged by Kosishka over there. So we developed it. So we are looking at all types simultaneous. And if there is a duplication, obviously you also have to see an increase in read depth. So we are plugging in read depth information. If the read depth is also increased, it is more likely an inverted duplication. So which actually helped in comparison to other tools that do not look at this types of variance. So these are only for inversions, and actually our area under the curve is improved with respect to others. Okay, so I'm already out of time, so I'll just go very, very quickly. So we have challenges with next-gen sequencing, let's just skip those old days, new days. So more recently, the pulse clone sequencing was developed to help with some of those problems, which is, again, a dead technology, but it was replaced by linked read sequencing. So I'll just go to the linked reads. So let's say we have a dense solution of large molecules, and then we dilute them into a bunch of pools, millions of pools, randomly. And each one of those pools is going to contain small number of molecules, 2 to 5, on the average 30 maximum. And then we barcode them, and then we sequence with Illumina at very, very low sequence coverage, 0.1x sequence coverage, but the actual genome coverage is going to be about 80x. And we know that they are likely coming from the same molecule, right? So yes, we did have PCS, but it is dead now. We use linked read sequencing now from 10x platform. So this is what happens when you have an inversion. So let's say this part is inverted, and then you have those molecules coming from outside, from inside, and from the junctions of these inversion. When you map them, these are going to map normally, and these are going to be split. So now it's going to be something similar to the split read, but we call them split molecules. And you can also have the extra read pair information directly from those inversions. So this is the split molecule signature. You have this haplotype inverted. You have 2 molecules coming from both junctions of the inversion. Ideally, they will be colored differently. So they are coming from different pools. And then when you look at their contiguity in mapping, you are going to see a split in contiguity. So if you see this, you are we just detect them as a possible split molecule, and then we cluster them again in a similar fashion with variation until if they agree on something, they are put in the same cluster. So basically, we applied that to 10x Genomics last year. It worked with this trio. So we are only looking for large inversions, in this case, megabase to 5 megabase range. And in this genome, on the 878 genome, 3 those large inversions were previously validated. And we could find all of them. We found an extra one, and then we confirmed it ourselves. And in the case of the father, we have some chromosome Y inversions that are also reported in the literature. So this is how the validation looks like. So this is a fish image for those that have never seen a fish image. Basically, we put labeled molecules into the cell, and this is a heterozygous inversion. So the order of those molecules are changed, right? So blue, green, red, and we have green, blue, red. So this validates our inversion. You can come up with similar formulations for other types of SV, and there are, by the way, I had a slide over there. So there are other tools such as Long Ranger, Neighbor, and Grok SVS that use similar strategies. More recently, we have Valor 2, which can detect duplications and inverted duplications, and Fatih, who developed it is among you that you can talk to him about. So in summary, well-integrated misaligned complex SV are hard to detect. They actually cause false positives in easier structural variation. Short read lengths make it harder due to the mapping ambiguity, but long reads help. It reduces the ambiguity. Long-range information helps. Now you can use that 30 to 50 kb information to more accurately map your reads or cluster them. And additional data such as optical mapping and Hi-C also helps. And finally, another conclusion from this talk: SNPs are boring. I'd like to thank my group and so on. Thank you.
Questions?
I have a question
Yeah.
about parities.
Mm-hmm.
So this inversion, genomic rearrangement detection from short read data.
Mm-hmm.
As I understand, in a normal case, when you do DNA sequence, when you want to make fragments as equal from the size as possible,
Yes.
not to introduce the bias, but this will introduce a bias for your detection of rearrangement. So you would, in your case, you would like to allow a big range of sizes.
No, but okay, so we are talking about now, we have Feridun's DNA, right? It is already rearranged with respect to the reference. And then we are using the size selection on Feridun's DNA. But then we are mapping those reads back to the reference genome. So they are all going to be about 500 base pairs in Feridun's DNA. But in comparison to the reference, you are going to have all those discrepancies in mapping. So
But you would lose, for example, if you, especially on this slide, you would probably miss,
which one?
Yeah.
This one.
Like the green case.
Uh-huh.
Okay, this is the reference. Sometimes when it's longer or shorter in the new genome.
Yeah.
you would- And as I mentioned, if you look at 1,000 genome data, they were all sequenced already with a very
Yes.
equal number of size left. It doesn't make it harder to find new things,
Well, I mean, yes. So in the beginning, I said that the size of the variants that you can find is going to be dependent on the fragment length. So ideally, you want bigger, but you are not always, getting bigger, fragments. Right? But again, those blue bars are about 500 in Feridun, but they are mapping differently in the reference because of the variant. So you can still find those. But yes, so the larger ones you have is going to help you, find more, larger variants. So these work, better for relatively smaller ones. Yes.
Another question? In the very back.
Sergey?
Yeah, great topic. So I was just wondering, even though the long reads are taking over, but what are the key properties of the short reads kind of to successful detection of structural variants? Is it like the in-source size distribution, or like the coverage, or like uniformity of the coverage? Do you have a feeling like for example, if you make your distribution of the in-source sizes like super high, and then you only relying on the split reads, can you do still a good job? Those kind of some insights.
Well, okay, it is all of the above.
That,
It is all of the above. We want the distribution to be tight. We want the molecules to be large, which they are not in the case, and we want the reads to be long, right? So obviously, in the first days of the 1,000 genomes, this didn't happen. So now it is way better. You can still rely on split reads. So with MySeq data, for example, now you have long fragments and longer sequences, which they actually overlap, and now you can do a small assembly of those read pairs into the 400, 500 base pairs, and then do split read on it. But again, it's complicated with the repeats in the genome. So it is all of the above, but let's talk to Mark about it on Thursday with long reads.
What things bigger again?